here and share this one. So hopefully you can see the correct thing now.
Yes, it's the right one.
Good, thank you. I'm going to pin myself, and that's it. Okay, so we will do the last little bit of this lecture. And I will explain to you now how a spinning disk confocal works and also light sheet systems. So a spinning disk system is basically a camera-based system like wide field, but with pinholes. So we have our same sample as before. We still have the inverted system. So we have the objective pointed up. Now there is no light on the sample. And we can use, we use lasers. And we have the dichroic mirror, so we have no excitation system filters normally because we use lasers, and this light goes through, so you see it's not focused, defocused through the dichroic mirror, and it reaches the pinhole. So the excitation light is going to go through the pinhole, and the pinhole is going to make it so that, so the pinhole moves. Turns very fast, really fast, much faster than the camera can image. And so it scans basically the all these little holes allow the light to go through, and that makes lots of small spots of excitation on the sample. And the pinhole that turns very fast is going to scan all these little spots of excitation on the sample. And of course, the sample starts fluorescing. And when the sample fluoresces, we have the emission coming back. And since we have lots of little spots, then we have also lots of little spots in the excitation. We can have up to 1,000 spots, or I think even more nowadays. Yeah, so thousands of spots, let's say, that of illumination and emission at once. But here we have the objective is focused on one particular plane. So this is the objective focal plane. And we also have the pinhole focal plane, and they are conjugated. And because of that, we have two conjugated focal planes, and therefore this is a true confocal. So a spinning disk confocal is a true confocal. Basically, it works the same way as a single point confocal. So the light that comes onto the sample is going to excite fluorophores at the plane of interest, but also above and below the plane of interest, and therefore they will fluoresce, and all this fluorescence comes back. But thanks to the pinhole, we eliminate everything that is out of focus. And this emission light is going to go to the dichroic mirror, which will send it to the emission filter. And we have then a camera. The camera chip has millions of pixels. And so we record an image, everything at once, like instantaneously. And if we look up at this image, we have also an XY coordinate and an intensity in each pixel. And this time the camera records both the XY coordinate and the intensity. So a spinning disk confocal is a true confocal. It's fast because it's a camera-based system. And we illuminate many XY spots in the sample, and we have an illumination that also illuminates in Z, and therefore we excite also above and below the focal plane. But the out of focus light that is emitted by the fluorophores that are above and below the focal plane are eliminated by the pinholes exactly in the same way that I was telling you, by basically only allowing the main lobe of the point spread function to go through the pinholes. And then the camera records the XY of each pixel and the intensity. And the excitation light is scanned on the sample, and then the emitted light is also scanned afterwards on the camera. So it's a little bit difficult to conceptualize, but I will show you an example that may help you understand. So this is another type of spinning disk confocal. The principle is exactly the same, but in this type, we have two disks. And the first disk is micro lenses, and they will help focus the light. So, but otherwise the principle is exactly the same. So we have the excitation light, which is defocused, and in here, these lenses will focus the light into lots of spots of light that go through the dichroic mirror and are sent to the sample. So this is an upright system because the objective is above. And then the sample fluoresces and the emission light comes back through the objective, as always for fluorescence. Both the excitation light and the emission light go through the objective. And then we eliminate the autofocus light with the pinholes. We reach the dichroic mirror, which sends the emitted light to the emission filter and scans it on the camera. So this is just another type of spinning disk with double disk, but the principle is exactly the same.
Can I quickly ask something?
Yes.
So the emission light, when the excitation and the emission light, they both go through pinholes.
Yes.
It's the same disk of pinhole, or how do you say?
Yes, it's the same disk, yeah.
And is it also the same pinholes?
Yes. So it's basically both the excitation light and the emission light go through this disk.
Mm-hmm.
Yeah. So, but this disk is only there to basically focus the light, but you can also avoid this one. So this is just two types of spinning disk confocals, one with an extra disk that focuses the light, but the part that removes the autofocal plane, the autofocus light, is this pinhole. So it's both the excitation and the emission go through the same disk.
Okay, thank you.
And now, how does that work? Why is it that we do not see these holes? Well, this is all about how fast the disk spins. So we look at this poor little guy there, and he's interested. He starts running. And what you can see is that when he runs very, very fast, we do not see the lines in here. And that is because he goes so fast, and our eyes, but then some accidents clearly can happen afterwards. But when he goes very fast, and our eyes and the camera that recorded this film cannot record the disc, or these lines go so much faster than the camera can record, that they actually disappear. And it's the same thing if you think about it when you cycle. When you cycle fast, you will not see the spokes in the wheel. It looks like the wheel is sort of transparent. And if you see the same effect, for example, in a car wheel, when it starts driving, you will get some optical illusions where you think that the wheel is going backwards. All these type of things are helicopter. You do not see, you will see the sky through the helicopter spade, or what it's called, I forgot the name. And that is because it goes so much faster than your brain can record. So it's all a question of speed of the recording compared to the speed of the spinning disk. And the spinning disk spins very fast, and therefore the camera sees through the disk. And so the image is recorded completely instantaneously. So what are the advantages of single point confocals? We have a flexibility of the compared to single point confocals. What are the advantages of single point confocals compared to a multi-point confocal? Because we can say, okay, the multi-point confocal is confocal, so we have an optical sectioning. So why would we ever use a single point confocal? Well, the single point confocals have more flexibility. So we have a flexibility of the scanned region. So we can decide any type of area and shape that we want to image on a single point confocal. On the camera-based systems, we're quite stuck. So we can make a rectangle, for example. We can crop the camera chip and say, okay, I'm going to image a rectangle or a square, but not much more than this. On the single point confocal, we can even have a single point. We can only image a single point, and that allows us to have some special techniques, let's say, that we can use, like FRAP and FCS and things like this, that can be used when we use a single point or a small shape, just a small area. So the flexibility in the scan region is higher for single point confocals. Another one is the flexibility of illumination. So on the single point confocal, the laser light is highly concentrated in a very tiny spot. So the power of the single point confocal is very high because of that, much more than on any other system where the laser light is not focused. And that means first that we have a very high power, and also we have clearly a high bleaching and photodamaging for live samples. But this is a very useful technique when we require a very high power, for example, for FRAP, when we want to do photo bleaching and photo activation of a small region, and also for STED, for example, where we can focus the light into one spot and then eliminate the rest of the light just around that spot to make the spot even smaller. So the power can be an advantage on the single point confocals. We also have, on the single point confocal, we also have this flexibility of the pinhole. So we will discuss about the pinhole size and what is the advantage of closing and opening the pinhole size. But basically, the pinhole size should be matched to the wavelength that you're imaging and also to the objective numerical aperture. Do you remember we said that the numerical aperture of the objective is basically, we will talk about this more tomorrow, but basically it is related to how much light can come into the objective and also the quality of the light that comes into the objective. So all this, the pinhole should be matched to the wavelength and the numerical aperture. And on a single point confocal, we can change the pinhole size to whatever we want. But on a multi-point confocal, we could have maybe 2 discs, but not much more than that. So it's quite often that on a multi-point confocal, we image with an imperfect pinhole size. We also have on a single point confocal the flexibility of the pixel size. And that is because I've told you that on a single point confocal, the pixel size is actually how much we jump from we image from one spot, then we go to the second spot to image. What is the distance between these spots? So we can decide ourselves how much we're going to jump. And that can be anything. So on a single point confocal, we have a very high flexibility of pixel size. We can choose whatever we want, and therefore we can choose the perfect pixel size for any wavelength and any objective numerical aperture. On the camera-based systems, we cannot do that. We are dependent on the camera sensor, basically the size of the pixels. And sometimes we can have little tricks to change the magnification, but mostly we are stuck to the size of the sensors on the camera. On a single point confocal, we also have the flexibility of emission filters because we can have a spectral detection. And if we use spectral detection, you remember we can open and close the filters, make them wider or narrower, and this is very powerful. So on the camera-based system, we cannot do that. So basically, the advantages of a single point confocal are the flexibility of changing all the settings. It also means that it's a little bit more complex to set things. So there are also tricks, as I said before, that can help us. Single point confocal can be faster if we use these little tricks, bidirectional scanning, resonance scanning, and several detectors, and even spectral unmixing. And we can add a little bit of flexibility for camera-based systems by cropping or having extra lenses or binning or having another pinhole disk or something like this, or having several cameras. But this is anyway limited compared to a single point confocal. So again, it is important if you use a single point confocal that you consider that you should use this flexibility and set your settings in the best way. If you use a single point confocal always with the same scan area, pixel size, and pinhole size, you're definitely not using your confocal in the correct way. And you would be better off with a camera-based system. So now I'm going to just show a little thing about light path microscopes. They come with all sorts of different architectures, let's say. And so they look very different. So this one is the Zeiss light sheet. It's like a box and you don't really see what's going on. So it's a bit difficult when we have people who have this type of microscope coming to our course. They could also look like this if you build them on the breadboard. And there are some people who do that as well. So some microscopes look like this. Our microscope looks like this. This is a light sheet system as well. And there's this big thing that is hidden behind, so it's harder to see. But anyway, we will understand how your system works. So this is the Zeiss light sheet microscope. It's anyway, all light sheet microscopes are camera-based. So basically very fast, but low flexibility. And the specific thing that we have with light sheet system is that the excitation is perpendicular to the emission light path. So in all the microscope I spoke about so far, the excitation and the emission go through the objective, but it's only one objective. Whereas in the light sheet systems, we often have 2 or more objectives, and they are perpendicular to each other. So the excitation light comes from one objective, and it is scanned very fast in such a way that it makes like a sheet of light like this. Okay, so it's like one spot and it's like scanned very fast. So it makes like a sheet of light. And that means that it only excites the plane of interest. So now we have an optical section because we only excite the plane of interest. And that means that we don't need a pinhole because we do not have any autofocus light. So it's a great advantage to have this excitation light that is perpendicular to the emission light. And then we image whatever comes out of the sample on the other side, exactly like a wide field system. If you want to say it's a little bit like a wide field system, but the illumination is different. And therefore, we do not have any autofocus light coming in, emission light coming in. So we have optical sectioning without pinholes. And we have high Z contrast and high resolution. So this is a little movie that I'm playing because I think it's quite useful. It's from the Leica, actually the old way in Leica to have a light sheet. They have stopped that now, but I think that the little movie is very clear to how the light sheet is produced. So you can see here the excitation objective and then the emission objective. So this is, they use another trick where they use only one objective, but the principle is the same. So I think you will understand how the sheet is formed, which is what I would like. So now they're going to zoom in, and now you can see that the laser light is coming in and bouncing on these mirrors back and forth. And then they start scanning this laser line very, very, very fast. And so fast that you actually see a sheet. So again, it's the same thing as the little hamster there in this stuff. If it scans very fast, then you will not see that it's single lines. So now we have our sample and our sheet of light, and we're going to move the sample through the sheet. And as the sample moves through, it gets excited when it goes through the sheet of light, and only this light becomes collected by the camera. So this is the way we can get an optical section from a light sheet system. So I thought it was quite clear on this little movie. So the challenges with light sheet microscopy, why is it that not everybody uses light sheet microscopy then? Because we have an optical section and we're very fast. And we have very low bleaching because we do not bleach above and below the plane. So there's lots of advantages in light sheet microscopy, but there are also disadvantages. So there are some challenges. First, in general, for light sheet microscopy, we're imaging quite thick samples, and therefore we have to make them transparent. And we will talk about this during the clearing lecture next week. I think it's on Tuesday. So that is a little bit of a challenge. Once you have the protocol going, it's fine, but you have to work on the protocol first. Another little restriction with the light sheet microscope is that very often we have several objectives. And if we have several objectives, it means that it's much harder to actually mount our sample. We need to have access from both sides, excitation and emission, without blocking anything. So, for example, it's much harder to use a multi-well plate. In some light sheet microscope, it is possible, but it's actually, you need to access the sample for both sides. So you cannot have anything that hinders around the two objectives. Also, any imperfection in the sample may create some shadows. And so there are solutions for this. Different companies come up with different solutions for that. They rotate the sample or they swap the side, a little bit like what we've seen in the Leica, or our system has an airy beam, which allows us not to have shadows. But also, when we rotate the thing, we end up with several Z stacks, and then we have to fuse them together. So there are some difficulties inherent to light sheet microscopy, and many of them are linked to the fact that we image big samples on light sheet microscopes. We have a very good resolution in Z, But many light sheets, you have to compromise. It's either good Z resolution or a large field of view. Ours doesn't have that problem. So, and the convolution is requires that everything is a little bit perfect. So the clearing and the preparation of the sample preparation is really important and crucial. This is the reason why not everybody uses light sheet microscopy. It's because it's a little bit harder to prepare your sample, let's say, and that's the reason why. But it's a very powerful tool. So now I just want to play this little tiny movie of that has been acquired on a light sheet microscope, and I think it is really it's a very nice it's an embryo that is it stopped playing. Okay, here. So you can see the it's a Drosophila embryo that is developing, and because we only illuminate the plane of interest, we really do not have a lot of light toxicity. So light sheet systems are very good for live imaging. Because of this low phototoxicity, we can take like images in 3 dimensions, but still here. Okay, still have a low phototoxicity. So this is a little summary of what I've said. These 3 types of systems, single point confocal, multi-point confocal, and light sheet, all of them have optical sectioning capacity. So it means that we will only record the emitted light that comes from the plane of interest. So this is not the same for wide field, but these 3 have optical section capacity. And then the excitation and emission light path, for single point confocal, they go through the same objective. And for multi-point confocal, same thing. The excitation and the emission lights go through the same objective, but for light sheet is perpendicular. So we have often several objectives. And we record the light with detectors on single point confocals, but on multi-point confocals and light sheet, we use cameras. And the speed is therefore not very high on single point confocals because we have to image pixel by pixel, whereas the speed is very high on light sheet and multi-point confocals that are camera-based. And we can have a speed of 100 frames per second, so 100 images per second with like 3000 by 3000 pixels. So you can imagine on your point confocal, if you would image 3000 by 3000 pixels, how slow you would go. And it's not the case with a camera-based system. We can go 100 frames per second. The photon efficiency is quite low on a single point confocal, but it's very high with cameras, and especially with the light sheet system, where here we are throwing away quite a lot of light because we have pinholes anyway, but here we have no pinholes, so the photon efficiency is very, very high in there. And the photo bleaching is very high on single point confocals because the excitation is extremely focused. So it's a problem to image with bleaching and light phototoxicity. But on multi-point confocals, we have some bleaching of the plane above and below, the plane of interest. But on light sheet, we don't have that because we only excite the plane of interest. So this is very good on light sheet systems. And the XYZ resolution, as you can see, I wrote excellent on all of them. So we're going to discuss all this later. Does a light sheet system or does a camera-based system have the same XY resolution as a single point confocal? We will explore this together. But in the single point confocal is very flexible, so we can change the wavelength or the objective and just adjust everything, whereas on a camera-based system is not very flexible. And in Z resolution, we have an excellent and flexible Z resolution. And whereas on multi-point confocal and light sheet, it's also excellent on multi-point confocal, but not flexible. So, and in light sheet system, we often have a quite low Z resolution. It depends on the system, but it's anyway not flexible. So the flexibility of the single point confocal is definitely one of the pluses. that you have, whereas there's low flexibility on the camera-based systems. And if we want to image in semi-transparent samples, we get here scattering of both the excitation and the emission. And in here we have sometimes problem with the pinhole crosstalk, but in here we have less effect of the scattering because the excitation and the emission are perpendicular, so they don't multiply, let's say. So this is the summary for the advantages of each system with optical sectioning. I would like you to reflect a little bit and think about your own project. If you think about the way you image, do you use the flexibility? If you use a single point confocal, do you use the flexibility? Do you think that if you could get a system that has an optical sectioning power, would it be an advantage for your project to go faster? In microscopy, we always need to balance up everything, and speed is definitely an argument. If you're running a screen or something, or you have lots of samples, you don't want to spend lots of time on it. So reflect a little bit and try to figure out if you would be better off with another type of microscope compared to the one you have been using so far. So we will have after this the workshop that we will have tonight, I think at 5 or something like this, there will be a document called the summary of all light paths, which we will show, we will come up where you will see like some sort of diagram like this, and you will see, okay, light path for looking in the eyepiece with transmitted light, and this is the light path. And so you will have all this summary in case you need to actually visualize the thing. This comes up at 5, but first we're going to do the workshop. So this is a little diagram to show which microscope you should use when. So basically, sometimes when we have many samples, we'd like to go very fast. And so maybe a camera-based system is best. But if we have slightly thicker samples, we need to have an optical section. So either a single point confocal or a multi-point confocal are good. And if we have a flat mallow layer, we can pretty much use anything, because then we don't care about the optical sectioning so much. But then we have very large samples, then light sheet systems are definitely the best. And if we have dim samples, systems that do not have pinholes are best, because we are not throwing away any light. So white field system, if we can, or a light sheet system, very photon efficient systems. And then if we have lots of colors, single point confocal is great, because then we can use image lots of colors at the same time and maybe change the filters. And live samples are not good on single point confocal, so any other system is better because of the fact that the light is so focused on single point confocal. And for very fast event, we have to go really quick. So we want to have a wide field system for photon efficiency and speed. Right, so in general, you would want to choose, if you can, you would try a wide field system. And then your choice number 2 would be a spinning disk system. And then number 3 might be a single point confocal, and number 4, light sheet. And the reason why the light sheet is number 4 is because of sometimes the difficulty of preparing the sample and mounting it. So we're going to talk about all this afternoon in the workshop. And we will revise the light path for each type of microscope, either looking at transmitted light in the eyepiece, or looking at fluorescence in the eyepiece, or imaging transmitted light and fluorescence. That's it. This is not for today, so I will stop sharing. And I would like to know if